Hello, everyone. Namaskara. Today let us consider the topic dicot root. Please remember the idea to memorize the dicot root, monocot root, dicot stem, dicot leaf and monocot leaf is to make your own comfortable note. I have made a pretty decent note here. Please modify this or take it down as you like. Let's go layer by layer. Outermost layer would be the epiblemma. Now the prominent thing here is the epiblemma has root hairs. Now the type of root hairs is unicellular. Extremely important point, unicellular root hairs. Now the next layer would be the cortex. Okay, now these cortex, it is not just one layer, it is in layers. And there are the cells which make up the cortex is the parenchyma cells, which are thin walled, and the cells have spaces between them. So basically imagine cells like this. So the thin wall, there are a few spaces in between, and also they are parenchymatous and they are in layers. So I shouldn't forget that. Just draw like this and that would be your cortex. That's how you remember. I put a plus sign here so that we can remember there are the features. We should never forget the features because the thin can be made into thick, intercellular can be intracellular, and such things can be important in the exam. Now the innermost layer of the cortex would be the endodermis. And now this endodermis speciality is it has a layer of cells which is called the type of cells here, it is shaped like a barrel. Okay, but here the contradiction is there is no intercellular spaces and it is only a single layer. And another important or an interesting thing is the tangential and the radial walls, they have a water impermeable or water resistant covering, that is the material made out of suberin, that is called as Casparian strips. Okay, so note down suberin, the water impermeable or water resistant material called as suberin makes up the Casparian strips on the tangential and radial walls. Now that we are done with the cortex, let us move to the pericycle. Now this layer has few layers, true. It is made out of parenchyma, true, but it is thick walled. Please do not have the misconception that parenchyma cells are always thin walled. There are situations like here where it can be thick walled too. Now what is the speciality of this layer is that the secondary growth starts from here or it buds from here. Say the lateral or the vascular cambiums, it buds from here. Okay, the next layer would be the pith. Pith is small and inconspicuous. It is like, please refer the diagram in the textbook, it will make you understand easier. It's like a mass of cells accumulated in the center. It is small and inconspicuous, which is contradictory to monocot root. Next, we have the conjunctive tissue. This is not in center, it is actually between the xylem and phloem. So it will be placed between the xylem and phloem, which are the vascular bundles.